early on we'll leave the light on for like a month straight, which anybody who's ever kept fish knows you're not supposed to do that. They need a nighttime, but when they're so tiny, you can't turn the light off or on or they'll die. We don't vacuum the bottom of the tank for the first maybe month, because just being in there with a little clear tube will be enough stress to scare them and kill them. So the camera definitely would be.
Yeah, you don't normally hover at my shoulder when I'm doing stuff like this, but you were like, I feel comfortable with a movement about like this, and you do like a little gentle move of your hands, but not like this, and you move it a little bit more.
And even just like bumping into the side of the tank, that is like an earthquake to them, it's a lot.
It's like operation.
Exactly.
Don't touch the sides.
Hey there, Podcast Aquanauts. It's your host, Casey here. Judging by the behind-the-curtain metrics I can see as the host of this show, I can assume pretty safely that many of you listening out there in the Podcast Aquanaut Corps are either parents or friends with plenty of people who are parents. With that in mind, today's episode is likely to strike a chord with you because we'll be discussing the many trials and tribulations of raising small animals, in this case, freshwater and saltwater fish. As any parent can attest, baby animals can be fragile, and ensuring they survive to reach adulthood is a path fraught with challenges and obstacles. At times, it can even feel impossible. Now imagine how difficult that task becomes when you're talking about animals that are smaller in many cases than a grain of rice when they hatch, with equally tiny mouths and a disposition so fragile that simply startling them can be more than they can bear. In the forthcoming discussion, two of our aquarists will discuss the many layers of complication they had to navigate when learning how to raise fish from eggs collected from our exhibits, and why, ultimately, this arduous task is absolutely worth the effort. But, like a baby myself, I'm just babbling here. That's enough from me for now, don't you think? On with the podcast.
Hello, and thank you for joining us for another episode of the Podcast Aquatic, the official podcast of the Tennessee Aquarium. Today, I want to talk about raising new generations of animals here at the aquarium in-house, which is a really cool program that we're doing on both the saltwater and the freshwater side. And there are differences in those two worlds that we'll get into in a lot of detail probably in the forthcoming discussion. But before we get into that, let's go ahead and work our way around the table with some introductions. We are once again in the glorious brick wall-lined office of Tom Demas. You're our Vice President and Chief Husbandry and Exhibits Officer.
That's right. Good morning. How are you doing?
I'm wonderful. I'm just glad to have you back on the show. You're like becoming kind of a regular on the show. You've been on like 3 episodes already.
I think so, and it's fun. I love it. I always love it when Casey comes to the office and we do this. So thanks for being here. I knock on your door
with a big bag full of just mics and equipment and all kinds of stuff. Next to you and across catty corner, I guess you could say, from me is Kyle McFeeters. You're our lead aquarist, life support systems, but I think you have a different way of saying it.
Yeah, lead LSS aquarist.
Okay.
When you know what LSS means, you can say that.
Yeah.
But filtration essentially is LSS, we say.
Yeah, I think I've said last time you were on, I said you're in charge, or your help with the pipes that keep the fish alive.
Yeah, essentially.
Sure. Okay. And then next to you and across from me is Ben Stinger. Ben, you are our lead aquarist for the River Journey building.
Yeah, much simpler.
Yeah, yours is your job title at least is easier to explain. Again, your focus being on the River Journey building, you are going to be contributing a little bit more of the conversation, or a little bit more to this conversation, looking at it from the freshwater fish side, which is we're going to find out in the course of the conversation is something that we know a little bit more about. There's a lot more history of raising freshwater fish than there is saltwater fish. And Kyle, you're over in the Ocean Journey building a lot. A lot of your responsibilities happen to be for exhibits that are in that building, like the Stingray Bay Touch Tank. You do work in Secret Reef as well, but in this particular case, I'm brought you on because you are one of the aquarists who has helped to kind of found and has seen through over the intervening years our effort to raise marine fish, so saltwater fish. So we'll compare and contrast here in a little bit, and then Tom, you're here because just your expertise is always welcome. So let's sort out terms first. I never know when we're talking, this is maybe just a blind spot in my own awareness of biology and the terminology associated with biology, but when you're talking about baby fish, do you say baby fish, larval fish, fry? What do you call them? And are there distinctions between those terms that make them all accurate, but just at different stages of development?
I think what you said last, that does make the most sense, that it is terms we use not literally interchangeably, we will use them throughout their development. Larval, we discussed the kind of broadcast spawners from the Secret Reef, We'll call them larval when they're first, coming out of the egg. They're super tiny. They drift on the current. They're planktonic. They don't look like a fish at all. Then they morph into what you would call a fry. But you can say baby fish anytime you want.
Okay. So like once they're recognizably a fish, that's at the point at which you would consider them fry?
Yeah, they have what's called flexion. I don't know if that's for everything. We talk about it with our saltwater larval fish. It's essentially when they gain a tail almost.
Okay.
It's kind of an easy way to describe it. And again, this is on like a super tiny level. But they go from just drifting and they can't do anything, and then they have this change where they go through flexion and they're more of a fish.
Okay. All right. So like when they come of fishhood, that's when
they're still very tiny.
Okay. And the size difference is probably something that we'll get into again a little bit later, but a phrase that you used a second ago that I think probably warrants at least some revisiting is broadcast spawning. So when you say broadcast spawning, what do you mean exactly? What activity are you talking about?
So we talk about saltwater fish being, recently raised. That's not entirely true. Lots of fish do spawn on a substrate. They make a nest. They guard the eggs. They grow for a period of time, maybe a month, and they hatch out as a big baby fish. And
it's a relative term.
Yes. Those have been raised, like clownfish, maybe 40 or 50 years. People have been raising clownfish. You can do them at home if you want. The ones that are kind of new to us, new to science, is these broadcast spawners. So again, fish that live in a big school in the wild, and just one night they're broadcasting sperm and eggs into the water. Those meet up and become fertilized eggs, and that's what we're trying to collect.
Okay. And we're specifically talking about broadcast spawning happening in the Secret Reef, which that comes up a lot because it's our largest exhibit at the aquarium at 618,000 gallons. So you're talking about that exhibit is home to, I don't think it's an exaggeration to say hundreds of fish. I mean, it's not thousands, right?
It is thousands.
Is it thousands?
Yeah.
Okay, so thousands of fish all living in one habitat together. The fact that this is happening, I guess, probably reflects that we're doing a pretty good job, Tom, of creating a natural environment for them. I think when you have behavior like this, to me at least, that suggests they're experiencing enough of the natural cycles and environmental stimuli to say, I should go through this part of my life as well.
Yeah, it's sort of completing the cycle of life. They're happy. That's a word a lot of people understand. Really what I mean by they're happy is we're meeting their needs. And so they're able to spawn. And what's really important about that is they're not just spawning, bad sperm and bad eggs. They're spawning viable sperm and eggs that are joining, and we're able to harvest those and raise the juveniles. So in essence, what that does is prove that the animals we have in our secret reef are thriving.
Yeah. So when you get reproduction, I mean, everyone loves baby animals at zoos and aquariums. And I think the best thing about a baby animal appearing at the aquarium, to me at least, is like you guys, the three of you who were actually involved in animal care, you guys are doing your job. You're doing a great job. You're making a good home for the animals. And to me, that's something that's always worth sharing with the public because it does show that their trust in us with these animals and their livelihood is well placed. So let's do kind of a quick overview of some of our efforts to raise fish. And I want to loop you in here, Ben. So on the freshwater side, how common is it for us to raise some of those freshwater fish? Kyle's already talked about the fact that when you're talking about marine species that are broadcast spawning, it's a relatively new effort. There's a lot of more history behind freshwater, raising freshwater fish. So what are we doing on the freshwater side?
Sure. I mean, it really just depends on the species. I think in our tropical department, we've had a lot of luck raising certain fish. But on the freshwater side, on the more temperate side of things, there's different factors involved than in the tropical side. It's not necessarily harder than one or the other. It's just different factors. And
like what? What kind of factors?
So, for instance, on the temperate side of things, these fish are used to seasonal changes, just like we are. So we're talking, winter, spring, summer, fall. So for them, that means changes in temperature. That means changes in rainfall, which can affect flow, pH, hardness, daylight length, food availability. All of these are different factors that play into their life cycle. Basically, they want to have babies when there's a lot of food and a lot of water. Yeah. So that usually means springtime, like early spring. So, the biggest challenge for us for reproducing temperate fish is mimicking those seasonal changes in a closed environment, an indoor closed environment.
Okay, so you said that we've had pretty good success on the tropical side. So when you say tropical, what are some examples of maybe exhibits at the aquarium that would be representative of tropical species where we've had some success?
Yeah, so think things like cichlids, tetras, rainbow fish, things like that, things that maybe you see in a pet store.
Okay, yeah.
And they also require different cues to breed, but some of them, the temperature might be the same. The factors that you have control over might be a little bit different.
Yeah, because like the closer you get to the equator, the less distinct the seasons are.
Correct.
Okay.
Yeah.
So
I think it's worth pointing out with the tropical fish that even if we aren't raising them, like the clownfish exhibit we have, I mentioned clownfish have been raised for maybe 50 years in human care. I didn't raise all those. There's companies that do it, and they're good at it, and I don't need to waste our time and effort because other people are already doing it with great success. So similarly, people aren't doing the fish that Ben's working with, so that's why they're trying to get that going. But a lot of people are doing the more tropical fish. So even if we didn't raise all those fish, they were raised in human care.
Okay.
Most of the fish in rivers of the world were raised in human care. So we have done it, we've dabbled in it, but we're not cranking out a bunch of fish because there are people doing it.
And that's important, I guess, to point out, because what some people might have concern about is, when you're getting fish that are sourced from someplace where they're raised in human care, that implies at least that you're minimizing an impact on wild populations, right?
Yeah,
Is that fair to say, Tom? I mean,
Yeah, that's very fair to say. There are a few examples where Project Piaba's one in South America where they go in and they get the neon tetras and they understand how they reproduce, and they're harvesting those tetras from the wild in a sustainable fashion, and they're selling them to create income. In exchange for that, they're not having to go out and cut down trees. So harvesting some animals from the wild in a sustainable fashion is a wonderful thing. The problem is, historically, our species hasn't done that really well. And the animals they're talking about being tropical are the ones folks have worked with for years and years and years, and we've had some of them in our fish tanks at home. The stuff Ben's really talking about is the stuff here in our backyard, in the mountain streams, things that maybe even are even legal to go into the pet trade, and certainly things that you would never see in the pet trade.
yeah. Well, and as we'll talk about, you're able to see some of the offspring for this tropical fish. In the Lake Nicaragua tank is, I think, a really great example of, you can fairly reliably go and see that exhibit, which is full of cichlids, which are very aggressive parents, good parents, and they breed a lot, or they seem to breed a lot, because every time I walk past that exhibit, it seems like there's always a little cloud of baby fish, with a mom or a dad guarding them and chasing off other fish, no matter how big they are, which is really cool to watch. I always love to point it out to guests when I'm at that exhibit, because they don't always see it, but once you know what to look for, it's kind of hard to miss. But yeah, the other things you're talking about, the marine fish culturing program that we're going to get into with Kyle, and the work that Ben is doing behind the scenes with some of our targeted species, our targeted freshwater species that are not being raised in human care, that's all happening kind of behind the scenes. And one thing that I wanted to point out in doing this podcast is to point out to people that by coming here, you are supporting some of these efforts to do things that are not being done everywhere. And that the work that's being done here is kind of helping to, on an institutional or industrial level for the zoological industry, to help to minimize our collective impact on wild populations by figuring this kind of stuff out. Ben, let's talk about, we've kind of alluded to it a couple times already. What are you working with right now, currently, that is a result of an in-house collection of eggs and raising larval fish?
Sure. So we're primarily working with the large stream exhibit in our Ridges to Rivers gallery. That was an exhibit that we, custom built to be able to mimic not only seasonal changes, but also different habitats so that we could accommodate a large number of species, because Tennessee has a massive amount of diversity. I'm sure you've talked about this loads already, but
You did actually. You on our one of the first episodes where we did a deep dive into this exact exhibit. You talked about it.
Sure. So we're working with, I think it's around 26 species in there. And so we were fortunate. Our main goal was obviously, as always, to make sure we have happy, healthy fish. And we thought that they would really enjoy living in a system that had all these different places they could go and travel and flows. And then we were also hoping that we would get the seasonal breeding colors that I know you've personally seen out in the wild, because we thought that would be great for our guests to see and would hopefully maybe bridge, help them make a connection with these animals that are living so close by.
People like pretty animals.
People like pretty animals, and we have some, fortunately. So that was another goal of ours. And then we were able to achieve that, and that was really wonderful, and we were all ecstatic. But then as the breeding season went on, we started to see not only breeding activity and nest building, but also we started to see eggs on exhibit. And so then it was like, okay, well, as aquarists, that's immediately where your mind goes is, can we get them out and can we raise them?
I want to take just a moment to pause, because we've talked about the point up to the point where the eggs appear. So in that past episode, we had talked about the spawning event the spawning event on a communal level had happened, and the excitement on everybody's minds when that happened throughout the entire aquarium was pretty immense. And I don't think I've talked to you about the next stage in this. So seeing the breeding colors and seeing that breeding activity is one thing, but once you saw eggs, like what was that like for you? Just given that our ambitions going into this were to create a true to wild habitat that you mentioned, where we would see some of these natural interactions
can
and
I
natural
add
behaviors.
something?
That's
I'm
like the
quoting
ultimate culmination
Ben.
of that ambition.
we were
or at least
talking
it seems that
about
way to me.
how
Yeah,
exciting
it was something
when
that
we built
I
this exhibit,
personally
this was our
hoped
goal.
for, but
That
you
we
never
could get
know
these
until
fish
you actually
to
build the
breed in
exhibit.
captivity,
So,
and we didn't know if we would get there, but we were hopeful.
seeing
we know if
the
anyone
breeding
could,
event
these
and
guys could
seeing,
do it.
but
when we were talking about it when it
for
happened,
everyone involved.
Not only
Please
were
do.
we very excited, but Ben's words were, "Now the real work starts." So just, everyone keep in mind all the work that went into designing and building this functioning exhibit that mimics nature well enough to get these streamfish to reproduce on exhibit, which to our knowledge has never happened anywhere before with fish that have these types of requirements. That really was just to get to the part where we could start to learn about how to identify eggs, how to get them to hatch, how to raise the fish. It's about sustainability, and now we're just at the beginning of being able to learn the part we really wanted to learn.
Which is that's got to be exciting.
Should I provide a little bit of context on why, like how the different things that needed to happen?
Yeah.
So just real briefly, because I think I've talked about this before, but so mainly when we talk about these fish, we're talking about things in the minnow family, shiner family, Cyprinidae. And so there are a couple different species that build nest or dig pits in order to lay their eggs. River chubs usually build nest, which is they pick up rocks, they build a mound of rocks. Stone rollers will do the opposite. They'll kind of dig a pit with their mouths. And these mounds or pits are great homes for eggs because they provide safety from predation. They provide oxygenated water. These structures are also really popular with a lot of other different species of minnows. And that's kind of where we got really excited because it was kind of this two-factor authentication of breeding. You needed the river chubs to breed, and then you needed the other stuff to breed. So
That may be the first time I've ever heard somebody say two-factor authentication in a positive
yeah,
light.
exactly.
Anyway, continue. Sorry.
And so that was what made it so special was you needed this key to sort of unlock the potential for all these other species like saffron shiners, Tennessee shiners, all the really bright, colorful ones. And so that was what was really exciting. They don't necessarily do all that without those initial species.
Yeah.
So that's what made it so exciting.
So, I mean, Tom pointed out that you also said that this is when the hard work begins. So how is that work going?
Yeah.
What are we learning, and how successful have we been?
Yeah, so once we did see the eggs, like I said, we immediately went to, well, what do we do now? And so I know that Tenacity, our conservation institute, has had success breeding native fish as well. So I spent some time over there and learned how they collected eggs. And then we brought some of those methods to the exhibit. And basically, if you've had a home aquarium, you have a gravel vacuum. We just have a very large version of a gravel vacuum. It's about 4 feet long.
I've seen you use it, actually. It is pretty enormous.
It's a little unwieldy.
You're usually diving when I see you use it.
Yeah. And we basically just vacuumed the river chub mound and sucked up the eggs, put them into a bucket, and this is the part where we had to, use a little bit of science, was we tried to take notes on go look at the eggs under a scope, take measurements, take photos, and just kind of get an idea of what the eggs look like. Because there's not a lot of information out there on these eggs. We've looked. So step one was just getting as much information as we can. And then we kind of separate the eggs by what they look like and then raise them separately so we can see what they grow into. We put them into jars with aeration. They only take a couple days to hatch. They're pretty quick. And then we take the larva fry and separate them and put them into bigger tanks and then try to grow them out and see what they are. And it takes a long time before they get to the point where we can even make a guess as to what they are.
When you say a long time, like how are we talking? Days, weeks, months?
I mean, for the recent ones that I've done, it's been, I would say, a couple months.
Okay.
Yeah.
Now, you mentioned that when river chubs make these mounds, that they are popular with all these piggybacking species.
Yes.
So when you get this just collection of eggs, how many species are we talking about? I mean, because you said 26 for the whole exhibit. Is every single species laying on top of this mound? I mean, how many species do we get out of this one collection?
I mean, we've seen a lot over the nest spawning, but we're trying to be very limited in what we take just because we have a limited amount of space right now. So we try to just take a little bit because we're just trying to learn now. We're not really going into full scale production or anything.
Okay.
But yeah, we've raised a lot of river chubs, so we know what river chub eggs look like.
Okay.
And we've seen Tennessee shiners, which was probably the most exciting one because that was one that has to utilize the nest. And then most recently we had a Tennessee dace fry that we successfully raised up. So
very cool.
And I know there's other ones in there. We just have to be, a little selective right now. But we've already learned a lot.
So right now it's almost as much about kind of developing the methodology as it is the actual production of new babies. Is that fair to say we're figuring things out?
We've made it up as we've gone along a lot of the way. Yeah.
The Tennessee Aquarium's podcast Aquatic is made possible by support from the Tennessee Conservationist, the official magazine of Tennessee State Parks. Each issue is filled with fascinating stories about Tennessee's native species, rich history, and unique culture. It's also your guide to exploring state parks and planning your next outdoor adventure. Subscribe today at tnconservationist.org. Tom, is it fair to describe these or characterize these as pioneering efforts? I don't want to claim too many superlatives on this because I don't want to say we're the first one or it's unprecedented because I don't think that's true. But are we on the leading edge of this?
Yeah, I would say we're definitely on the leading edge. I think what Ben is doing with this stream exhibit and these eggs, we may cross a line where we're the first one. I don't think we have yet. I'm not really sure.
Some furrowed brows around the table here.
Yeah, I do know not many people are working with these species, right? CFI is working
Conservation Fisheries Incorporated up in Knoxville.
They're working with some of the same regionally native species. So again, I don't want to say we've been the first, but what I can tell you is what we're learning is directly applicable to other similar species. So even though maybe we're not the first one to do it, we're working out the protocols, we're working out the how-tos that will help either us later or someone be the first to do some species.
Yeah.
And I'll give a quick anecdote if it doesn't take too much time. I remember Ben was giving a presentation about this exhibit because we had the initial spawning, and he was talking, I believe it was actually during the presentation, about how all the work they had done going out and looking at the natural habitat, studying the natural history of the animal to be able to properly replicate it. And his comment was you'd snorkel up, and they'd be out in the streams in the cold water snorkeling, trying to learn. And he said you'd snorkel up and see this bundle of rocks, and there's some algae on them and maybe a piece of driftwood there, and how beautiful it is, and think, man, that's what I want in the aquarium. It looks so good. And then you look over and there's this ugly bare rock and all the fish are fighting over it. So again, my point is when you're starting to do these things, it's not what looks like makes sense all the time. You got to figure out the triggers. You got to figure out the preferences. And it's really, really about understanding the natural history of the animal to get them to reproduce. Then the stage they're in now, learning what that natural history is as they're developing. And when you can't open a book or call somebody on what that information is, you've got to figure it out yourself.
Yeah, we've talked about within the zoological community in past episodes. We've talked about how there's so much intercommunication between institutions. There's not like some people might look at a zoo and think that you wouldn't want to talk to another zoo because that's your competitor. And that, at least in my experience, is just not at all the case. That there really is a reliance on the entire communal pool of knowledge when it comes to things like this, whether it's animal care or, and I guess this is animal care, but this particular aspect of animal care, that what one person knows, they want to share that information. We want to share that because I guess in this particular case, helping others to follow in our footsteps, if we are leading the pack in this, will ultimately expand or multiply our ability to minimize the impact, right? So the more institutions that are doing stuff like this, the fewer fish are having to come out of a natural system.
Yeah, absolutely. And I think the collaboration between the animal people at the different facilities often facilitates success with the animals we have in our zoos and aquariums.
Yeah. And Ben, are you talking to other institutions about this work? Are they expressing interest in it? Do they have you been approached to share this knowledge yet, or are we still at an early enough stage that people outside the building don't even know that it's happening?
There has been a little bit of interest, after we did our AZA presentation, but it's still pretty early in the process.
Yeah.
So, we're always happy to share whatever we know, but I feel like we're still in that very early stage where we're just trying to get data and then figure out what to do with it.
Okay.
But the fact that it's moving along at all is really exciting.
So, Karen, you raised your hand like you were.
Well, I didn't want to. I wanted to interject at an appropriate time. So Ben and I work together, but we really have like parallel paths. I don't see him, but maybe every couple of weeks. Like we don't collaborate on much stuff. So I'm hearing about this like now. I know the work they were doing. I didn't realize like the pioneering, how the successes they've been having has been going. We just hadn't talked about it.
It's dangerous to have too many lead aquarists in one space at a time. Like we have to limit this conversation to an hour because otherwise bad things will happen.
We can get on tangents. So just like a reaction to what all they're saying, I'm hearing this right now for the first time. And I gave a talk about our culturing, the saltwater culturing. And I mentioned the quote that's like, we're all standing on the shoulders of giants. I had this huge foundation of no knowledge given to me and said, hey, this is what we've tried that works. Doesn't work for that species? Okay, I'll tweak it a little. But I had stuff to go off. So for them to be doing this from zero and having success is just, that's amazing.
Yeah, I wanted to kind of pull you in actually along the same lines because when Ben was telling his story, I was hearing a lot of, like you said, parallels to discussions that I know you and I have had, and one of our past aquarists, Tasha, who the two of you were the ones who kind of started this effort to raise our eggs out of the secret reef back, I think it was early 2017 or early 2018. So at this point, you've been trying this for about 7 years. And I remember talking back in the day, and you said, there were other places that were doing it. It was still, I think, fairly new. I don't think it's hard. That's a mischaracterization to say that it's not something a lot of people are doing. There were some people trying it, but not a lot of people. There were definitely felt like a lot of just trying to figure stuff out. And I guess that's a lot of your job when it comes to stuff like this.
My takeaway for that was not to derail and to talk about saltwater fish, which is all I want to talk about generally. But my point was that we had a foundation that we could build off, just like handed to us. We had to figure out, and we are still learning more as time goes on. But to start essentially from zero, as they're doing, to try to simulate that habitat. Okay, we got to spawn. Okay, now what? Like I again hearing it right now for the first time, I'm just really like excited about that and blown away by it. That's awesome.
Like, do you feel daunted like by proxy by how?
Well, no, because I'm coming in that they've already done it. So I'm like, oh, thank goodness.
If it makes you feel any better, we stolen a lot of your copepods to raise those fry.
I know y'all use some.
We couldn't have done it without you.
I was going to ask too, like if they were so little that they needed much of that.
Oh no, we stole your copepods.
Yeah, okay. So this is where I think it makes sense for us to talk about the distinction between a lot of the eggs that you're working with, Kyle, and the eggs that are maybe more typical of freshwater fish, because we've talked about this in the past for other things, you and I, about there being some key differences between the broadcast spawning species eggs versus a lot of freshwater species eggs, and why a lot of ways that those differences affect the difficulty of raising those fish. So let's compare and contrast.
So if I make broad statements like that, I'm talking about those cichlids that you just show up and there's a bunch of baby fish thriving in the tank and you haven't done anything. You haven't even fed them. Those are the fish that they protect their eggs. They grow big before they hatch. That's what I consider simple or easy, again, because you haven't done anything and there's suddenly a bunch more fish in the tank. The fish that we're collecting the eggs from, they're spawning one night and the eggs hatch the next night. So they go from zero to a larval fish in 24 hours, essentially. And it sounds like Ben's aren't much longer than that. So just hatching so small that they need specialized food. That's the biggest hurdle.
Yeah, when you say small, can we give people some idea of how small we're talking about?
Maybe 1.5 millimeters.
Oh, well.
What about in American measurements?
1/32 of an inch.
Oh, yeah.
Let me interject here. Let me interject here. For years, these fish have been spawning in our exhibits, and we didn't know it.
It's nothing new, right?
Because you couldn't see it.
Yeah.
Right? They're so small, it looks like a little debris in the water. Oh, the water doesn't look great this morning. We need to do whatever to backwash our filters, whatever. Once you know and build a mechanism like Kyle has built to collect, and then can decant off all the water and look at what's left, then you can see, oh my gosh, those are eggs. So many of our guests for 30 years have probably walked by exhibits and maybe seen some eggs, and they were so small they didn't know they were seeing them. So that's what we're talking about.
So very, very small.
Wow.
You said
Very
1/32 of an inch. Like by the time you usually call me to say, hey, I've got some baby fish to look at, they must be pretty far along, because even when I see them, they're like the size of a grain of rice, it feels like.
Yeah, the video we did recently, I called you because I thought it'd be cool to put the camera in the water. And so I waited till they were maybe half an inch long because I was scared to kill them by putting the camera in the water with them. So you've done like top-down filming with them that are maybe, a grain of rice, quarter inch. But yeah, they start way smaller.
Okay.
Kyle, just expand on why putting the camera in the water may kill them and all those types of challenges you have to deal with.
It's not electrocuting them. Let's be clear about that.
Yeah, definitely not that. These fish at this stage are just so small and fragile. They're drifting on the current. The sun comes up, sun goes down, but that's about the only change that exists in their world when they're tiny in the wild. So in these tanks we're trying to raise them in, if the light suddenly turns on, that's enough stress to kill them. So early on we'll leave the light on for like a month straight, which anybody who's ever kept fish knows you're not supposed to do that. They need a nighttime, but when they're so tiny you can't turn the light off or on or they'll die. So same way, when we're working the tank, we don't vacuum the bottom of the tank for the first maybe month, because just being in there with a little clear tube will be enough stress to scare them and kill them. So the camera definitely would be.
Yeah, you were very on tenterhooks, I guess I could say, when I was there. Kind of, you don't normally hover at my shoulder when I'm doing stuff like this, but you felt like you were closer than normal to kind of see how I was doing stuff.
It was twofold. I felt pretty good that they were stable enough, but I was also trying to help you.
Yeah, and you were like, I feel comfortable with a movement about like this, and you do like a little gentle move of your hands, but not like this, and you move it a little bit more.
And even just like bumping into the side of the tank, that is like an earthquake to them, it's a lot.
It was like operation.
Exactly.
Don't touch the sides. Let's roll things back just a little bit, because we've talked about the fact that this started 7 years ago, and Tom mentioned that this was going on in the Secret Reef for years and years, and we just weren't aware of it. What was it that pushed us to the point where we decided to try and did we know that we figure out that those were eggs and then decide to try and raise them, or what was the impetus for us starting this project 7 years ago?
Tom came to me and was telling me about the program Dr. Andy Ryan was doing at Roger Williams University, whereas they basically got a grant to host aquarium aquarists to their aquaculture lab and show them us, what they're doing and the successes they've had and how it all worked. So we were able to attend that, and they're like, oh, your tank is how big? You have how many fish? Yeah, you're getting spawns in there. I'm like, oh, well, how do you know? They're like, just you are, for sure. So, coming home from that, putting an egg collector in, and every time you put it in, you get eggs.
What's an egg collector? Because Tom's already talked about this or alluded to this. How do we get the eggs?
So we built a device. Essentially, when these broadcast spawners spawn, the fertilized eggs become buoyant. And so we built a device that we can put in the water when we want to collect eggs, and it will skim the surface of the tank and collect some small fraction of the amount of eggs that are being put into the water.
Okay. All right, so you built this device. Talk about the first time that you collected and said, "Oh, holy cow, he was right."
Yeah, we didn't know what to expect. Again, they just said, "Oh, of course you've got a diverse group. You're getting spawning. Like you see 2 rabbits in the yard. In the spring, you'll have 5." It was like that kind of certainty to them. Like, of course you're getting spawning. And,
Idiot.
Right. That's how it was. Like, duh. And but Tom's saying that we never saw those are a bunch of eggs. Sometimes the tank was clearer than others. So you had to put the egg collector in and then actually get to see those tiny spheres on the screen of the egg collector. Like, oh, there really are a ton of eggs in here. Again, we're collecting just this tiny amount that can surface skim from that one little area, and we're getting a lot. So there's got to be so many every night.
Yeah, the contrast of your story versus Ben's story is interesting, because without even trying, there was eggs spawning or eggs were appearing without us even realizing it, versus for you, Ben, I mean, the eggs appearing was like literally that was the end goal. That was like like I said, the culmination of all that effort and ambition and planning, and all of that led up to those eggs showing up, versus Kyle just had, I mean, are we talking hundreds of eggs, thousands of eggs?
We get hundreds in our little collector.
And how often do you collect?
Just when we have time and room for them. So when we want some fish to raise, pretty much anytime we collect, we'll get them.
Ugh, that's got to be frustrating. All that it's so easy for Kyle.
No, I mean I have a planted system with killifish, and I found a fry in the sump yesterday. So it happens with freshwater too. A lot of it is species specific, not necessarily salinity specific. Every species is different. We still have to crack the code on a lot of different species, and some native fish are very easy to breed. Some are really difficult. So you really kind of have to you can generalize, but every now and then you get species that are just very easy or very difficult, and you just haven't found the code to unlock it yet. So
when you say difficult, what would make a species more difficult versus another?
I mean, some species require the milt of other species breeding. Like we think so
for
a long time they thought
sperm,
that,
I guess.
Yeah. So for a long time they thought that you needed river chub milt to breed things like saffron shiners, which is, again, I mentioned 2-factor authentication. But so yeah, or just very specific water parameters that we haven't even thought of yet. Water conductivity, water hardness. I mean, they just, they just don't want to. I don't know. There's a lot of species that we still haven't quite figured out, or we don't have the space to do it.
So given that there are so many different parameters that we don't know, like what's going to be the key to making this possible, I guess it really is ultimately to the benefit of this whole effort that baked into the design of this exhibit was the ability to have very fine granular control over a lot of different things, right?
Yeah, that's correct. We haven't had a system that was sort of that big and that, had variations in flow. Flow was really important for this exhibit, and I think it was one of the bigger keys in unlocking, the breeding cycle.
I don't know, it just sounds like a lot part of me, I guess, is just frustrated from afar for you, because that just sounds like, are you just up there throwing wads of spaghetti at the wall and just seeing what sticks? I don't know how you would figure a lot of this stuff out.
I mean, you can always look at the natural history of an animal, and that'll give you a lot of the data that you need. It's just a matter of, can you replicate that in a closed environment? And that's the tricky bit. And
I would just add, what factors might there be you're not considering? Right. So we were really lucky. We had the foresight for water flow, water depths, how much light, food, temperature, the rate at which the water moved. We designed this exhibit to do all that. And we got lucky that there wasn't some peculiar water quality parameter that we were unaware of that they needed. Had that been there, we would be looking for that needle in the haystack. So we were able to replicate it close enough to get to the success. And the other thing I'd like to say, thinking about the two tracks, the freshwater and the saltwater, Ben's journey has been figuring out what parameters do I need to get these fish to spawn. And now he's got to figure out, how do I raise them? While Kyle's journey has been, oh my God, these fish are spawning. How do we collect the eggs? And now that we have them, what do you feed them? How do you get them big enough they don't die if you turn the lights on?
Yeah.
So they're kind of two different tracks that have similar problems but different, resulting in a larger view being the same issue. Yeah. Hard to raise little fish.
Yeah, let's talk about the difficult. Anybody who's a parent knows that keeping any small animal alive is difficult. In the wild, no human baby would survive without parental oversight. I'm just convinced of it. And Ben, you're a dad, but
do your kids try to eat each other too?
There have been some bites, for sure. But what is it about raising baby fish in particular that is such a challenge? I mean, even once you get them out, what are the things that you're having to think about, things that they're sensitive to, that you're just having to think about in terms of their care requirements that make them inherently difficult to get across the finish line of reaching adulthood.
Yeah, and I don't claim to be an expert at this at all, but there is sort of this very funny window where they hatch out and they've got their yolk sack, and it's easy peasy because they've got their dinner there with them 24/7, and you're like, this is great, this is easy. And then the yolk sac goes away, and it's like, oh no, I have to feed them, and they're so small. And that's when things like copopods come in really handy. But that's the trickiest bit. There's that window about a week where it's just like, come on, buddy, we're gonna do our best. And then once they get a little bit bigger and they can eat 24-hour brine shrimp, and then we go back to it being relatively easy.
Yeah. And you mentioned copepods now a couple of times, just in case that's something that you're not familiar with. I wouldn't have been familiar with it if I didn't work here. That's just a very, very tiny crustacean, correct?
Exactly.
Yeah. There are some footage of that, of them from very up close that I think we were both pretty impressed by in the video that Kyle mentioned earlier. I don't know how recent it'll be by the time this episode comes out, but it'll be on our YouTube page if you want to look it up. Talking about the larval fish culturing. But yeah, they are very, very tiny copepods. And that was one of the big hurdles for you and for Tasha early on, as Tom alluded to earlier, is with fish that tiny, the food that you normally would feed like one of our other small animals still would be too big. So could talk about how we figured out ultimately that we were going to go with copepods, because there were even steps beyond that. It was like a layer. The onion just kept going in terms of complications and obstacles we had to overcome.
Ben mentioned getting onto brine shrimp, being able to graduate his baby fish into Artemia brine shrimp. Basically, when the brine shrimp, we hatch them every day, and when they're 24 hours old, they're the size of the adult, the biggest egg-laying copepods that we raise. So the fact our fish, the saltwater fish, generally takes maybe a month to be able to eat 24-hour brine shrimp. We knew we needed something much, much smaller. So not only can we take the copepods, the one we use is called parvocalinus, and there are smaller copepods species as well. We used that one. It was available. It's durable, so we liked that guy. And what we can do is sieve out the adults and only offer the baby copepods. So the adults are nearly as big as brine shrimp. The babies are 1/10 that size, so we can size sort the food we're going to give. Once you have the copepods established, they're pretty durable. They're pretty easy to raise. Their issue is raising their food. So just like the fish, the issue is raising the food for the copepods, which in our case is an algae, and the algae is just pretty sensitive.
Yeah, I remember when we first started talking about this being just. You were talking through like a complication, like, okay, we've got these fish that are kind of fragile. They've got very small mouths. What do you feed them? And I was like, yeah, that's a good question. What do you feed them? You're like, we feed them copepods. I'm like, oh, okay, that sounds complicated. You're like, but they're even smaller than that. What do you feed something that has a mouth that's like 1/10 the size of the one that you didn't know what to feed in the first place? And then the algae is its own complication, right? Because there was to raise the quality of algae that we needed, and I guess the amount that we needed, like there was also parameters that it needed. So talk about how do we get the algae going?
So the algae is obviously like the foundation for all of this, and its issue is it's very easily outcompeted. So anything contaminating your algae will take over, whether it's a different algae or yeast or bacteria. Anything getting in there will ruin it. So we just have to kind of make our process as sterile as we can. We actually will take reverse osmosis saltwater, which is the purest saltwater we make, but it's not sterile. So we take that nice saltwater and we will actually bleach it, and then we will neutralize that bleach so we know that water is super clean and pure because it's the reverse osmosis water. So we use that as the foundation for our algae. We will filter the air that goes in to bubble the algae down to 1/4 of a micron to be sure we're keeping anything out that would get in there and contaminate it. And then we do it 1 gallon at a time so that if something does go wrong, we don't lose all of it. So we still have something to feed our copepods, to feed our fish. You just lose 1/10 of what you're growing.
That's crazy. Well, one thing I like as we kind of wind this discussion down, because I feel like we're running out of time, first of all, but I do think that we've done a pretty good job of covering the challenges associated with both aspects of this and the fact that you guys are both doing this. One thing I like about this conversation that I don't know that I expected to come out of it is the fact that there's this kind of mutual admiration society kind of air to the room right now. Like you guys are all patting each other on the back, and Tom is patting both of you on the back. I would like to turn to you, Tom, and say, as you hear what they're talking about and the challenges that they've overcome, and looking at these projects from maybe a more strategic level, top-down view, what are your thoughts about why the success that we've achieved, the obstacles that we've overcome, but also in a broader sense, why is it important that we're doing things like this at the aquarium, and just in general, why aquariums and zoos should be involved in projects like this?
Yeah, well, there's enough of us on the planet that the planet's not going to get healthier and cleaner by itself. We as a species are going to have to step in and help. And if we're going to do that, we need people that are driven to do it, like Ben and Kyle. It seems like working with the larval marine fish at the Tennessee Aquarium and these larval stream fish at the Tennessee Aquarium, how are you going to save the planet with that? But I go back to what we talked about earlier, the processes that they're learning and figuring out and documenting. Those are the things that are going to pay dividends going forward. So why should we be doing it? One, hopefully it's exciting and motivating for them. Two, as we're able to do this, it takes pressure off of us needing wild animals. We can produce our own. Three, the knowledge we learn from this can be applied going forward, whether it's to work with other animals or for a larger purpose, aquaculture or something like that. And everything we can do to make a difference is additive. And if we're all involved, if all the zoos and aquariums are involved, we're going to get to an end goal quicker than if it's one or two. So the reasons it's positive are, and there's more, it goes on and on. The reasons it's positive are endless. And the work, it doesn't seem important. But at the end of the day, if these fish are thriving in the stream in nature, that's the same source water that we're drinking. So it all really goes back to that. What can we do to take care of our environment and to make sure that we're not polluting it and we're not destroying the things we rely on to survive? Our whole staff, but these guys especially, should be applauded for their diligence and effort. It's exciting and fun if you find this kind of thing exciting and fun, so it wouldn't appeal to everybody. But everybody who has something they like understands what it's like to go after it and stay after it and fail, and I'm getting close and it doesn't work, and they don't give up and they stay after it and they're successful. And they should be recognized and applauded for that.
I almost did a golf clap, but I didn't want it to sound trite or like I was being sarcastic. But I'm applauding inside. I think I do. I echo Tom's sentiment there that it's hard to do something that you feel like you're just making incremental progress, and it's just kind of a war of attrition. But I think, like he said, stalwartly pushing forward against all of that is ultimately just something that's very laudable. So, good for both of you.
Well, thank you. Thanks for helping make it happen.
If you have an idea for another episode of the podcast that you would like to have us cover, or a question maybe for Ben or Kyle or Tom, go ahead and send that to me at podcast@tnaqua.org. While you're online, leave us a review or a rating. Both of those are free and easy ways to support the show and help us with the algorithms so that more people can hear us and learn about these interesting topics. Well, hopefully they're interesting. And in the meantime, until next time, remember that every drop matters.